Hi everybody, welcome back to Principles of Organismal Biology. Last class we learned about species, how species originated, and the different mechanisms that they have to maintain isolation, reproductive isolation between these different species. We also learned about how species can evolve in the same place, that means in sympatry, or in different place, or separated in allopatry, right? Today, we're going to learn about the different methods and the tools that we as scientists use to make sense out of the different species. In other words, how different species relate to each other. That's what we do in phylogenetics, and we're going to be covering that today. But before we jump into that, I want to make a big announcement. So the first exam is coming, and next Friday, in September 18th, at noon. And for that, I have posted the study guide under Module 9. The exam is going to cover everything from Module 1 to Module 9, and that includes prokaryotes in today's class as well. You need to complete these modules before the exam. That's going to be very important. Today, we're going to learn about classifying species, how we classify species, and we're going to go through some of the binomial nomenclature, what that means, how we construct phylogenetic trees, and how we can read them, these different types, homologous and analogous, and these different type of clades, monophyletic, paraphyletic, and polyphyletic. From now, I want to say that what scientists really want to describe is the type of monophyletic group. So remember that monophyletic group is the one that we care about it. I want to start by explaining how we classify species. For example, if we think about the Greeks, we could think about how they were classifying things. And they basically had this classification in which they distinguish animals from plants, because animals run or move and plants do not move, right? That was the big difference. And they also have a classification for minerals. Linnaeus made some important contributions. So in the 18th century, Carl Linnaeus published this book, The Species Plantarum, in 1753. And just for you as reference, this is 1 century earlier than when Charles Darwin published The Origin of Species in 1859. So this is before Darwin, and Carl Linnaeus tried to classify a species based on resemblance. And we'll see that sometimes resemblance actually represents the evolutionary history, but sometimes it doesn't. So this is a tricky way to classify a species in some cases. And he also introduced a way to name a species. So classifying and naming a species. For the way that he proposed how we name a species, he used this binomial nomenclature. So bi, it's by two, and nomial, it's referring to the two components, the two parts, and the name, the scientific name. And this is an example. The first part of the name refers to the genus. And the genus is going to be a group of species that share some similarities, so they belong to the same genus. In this example, we have the genus Pinus. And what we want to highlight regarding the way that we write the genus is that we capitalize the first letter, like it's uppercase, and we also use italics to represent this. The scientific name is going to be in italics, and the scientific name is going to be compounded by the genus and the epithet. The epithet is going to be a word that is going to be unique for each species in that genus. If you remember the example of the pines that I studied in my lab, Pinus monophylla or monophylla, it's one species that the epithet is monophylla, and that means one mono, and phylla, it's the bundle, one leaf per bundle. And the whole name is going to then be compounded by two parts, genus and the epithet, and they're going to be in italics, and the genus is the only one that is going to have the uppercase. It's going to be important. The other part that Linnaeus contributed was this hierarchical classification, in which we're going to have increasingly inclusive categories. So if we start here, we have this Panthera pardus, that's the species, and then several species of the genus Panthera are going to be here. So several similar species are going to form the genus. Several genera is plural for genus, are going to form the family Felidae, and several families are going to form the order Carnivora. Several classes, several orders are going to form classes, and later phylum, and then we'll have the kingdom. Remember, we used to use the classification of kingdoms, not anymore, because there are some that are artificial. But then we have the domains in here, and then we have that if we use the kingdom Animalia, so these species belong to the kingdom Animalia, and this kingdom Animalia will belongs to the domain Eukarya. In the past, there was this mnemonics. King Philip came over from Great Spain, and you could add maybe something that starts with D. A student in the past added Dom. King Philip came over from Great Spain to represent the domain. So you see, now we have more increasingly inclusive categories. This is a phylogeny. Phylogeny is going to represent, it's a hypothesis of relationships, evolutionary relationships. This is Panthera pardus, the same that we were looking at before. All of these species in this tree are species from the genus Panthera, right here. So we have Panthera uncia, Panthera paleosienensis. All of the taxa that have a cross are extinct species. And then we have, for example, Panthera tigris over here. We have also another genus, Neophelis. This is a different species that doesn't belong to the genus Panthera. Panthera will be all of these. Okay, now, this is what we went through. That means that the family now, if we look at different families, if we look at different genus in this family, the family is Felidae, and we'll have this genus Panthera and the Neophilis that we were talking about before, and we have all of these different genera over here making this larger family. And then more inclusive group will be Carnivora, which has more. Now we have the family Felidae that we went through, but it also includes the family Mustelidae and the family Canidae. So your dogs are in this group around here, right? This is Carnivora, so they are species that eat meat. And we can see this hierarchical classification. So those are the two contributions that Carl Linnaeus had, and we are still using the binomial nomenclature, and somehow we use still this hierarchical classification. Two concepts that I want to introduce is the systematics and taxonomy, and they are sometimes interchangeable, but they have a slightly different, some differences. So in systematics, systematics is the study of kinds, similarities, and diversity of organisms and the relationships among them. This is going to be key for systematics. In systematics, we really want to establish the phylogenetic relationships or the evolutionary relationships. That means who's most closely related to another species, or what's the sister group of a species, if you want to say it that way. In taxonomy, in contrast, we're going to have, that's the theory and practice of identifying. So we are going to identify a species. We can describe the species. When you describe a new species, you find a new species and you have to describe it. If you describe it, then you have to name it. Otherwise, how people is going to refer to it. So you give it a name, and you also under taxonomy you classify organisms, and you can classify organisms without establishing the relationships. So that means that you can put all of the species that are pink in the pink box. but you don't care about if species A and B are most closely related than to species C. Hopefully you understand that. This is a cartoon of this concept of the tree of life. In this tree of life concepts, we really want to understand the phylogenetic relationships, the evolutionary relationships of all of these species on Earth. Yes, that 1.5 million species that have been documented, and not only those, but the ones that have not been documented and the ones that are extinct. We want to understand these evolutionary relationships of everything. In here, we have the origin of Earth, which we have said that it happened 4.5 billion years ago. And since then, we have had this speciation, gradual speciation at sometimes faster than gradual, but we have all of this diversity represented here in bacteria, and archaea, and eukarya. And this is eukaryotic center, this cartoon, because we have a lot more species now that we start documenting in bacteria and archaea. As I mentioned, early taxonomies classify species as either plants or animals, and later there was this classification of the 5 kingdoms. In these 5 kingdoms, we will have the kingdom Monera, which included all of the prokaryotes. And prokaryotes are going to be the ones that lack true nucleus, and those are what we call now as domain bacteria and archaea. And this, I want to emphasize that this kingdom Monera is an artificial group. It's not natural, and I'm going to explain more about it today and in the next module. Next, we have Protista, Plantae, Fungi, and Animalia in this 5 kingdom classification system. And we can see them in here. So Protista is another group that is also artificial, and we have abandoned this classification for this reason, because of prokaryotes and Protista are not a natural group. But Fungi, Plantae, and Animalia are still true or natural groups. Now, most recently, with the advance on some technologies, such as the sequencers, we are able to sequence genomes and sequence or just sequence pieces of genes, and we were able to determine that actually all of the groups in eukarya are more closely related than archaea with respect to bacteria. And that is the reason why we have separated and accepted that we actually can classify the species in 3 domains based on the evolutionary history. This is how this phylogeny looks. The domain Eukarya, it's right here, and it includes plants, fungi, animals, and it has all of these protists that are not grouped together, but they're more randomly, not randomly, but scattered in this tree, in this part of the tree. And then we have archaea, which is the sister group of eukarya, at least in this tree. And then we have the domain bacteria, that is the sister group of a lineage that carries both eukarya and archaea. And as I mentioned before, eukarya is the group that has a species with true nucleus of mitochondria, and it's going to also have chloroplast. And there is this theory proposed or followed by Lynn Margulis and elegantly articulated by Lynn Margulis. And what she said is that the chloroplast and the mitochondria are actually results of endosymbiosis. This is the serial endosymbiosis hypothesis, and now accepted as a theory because of all of the evidence that we have for this. And in this ancestral prokaryote, we have the DNA that lacks nucleus, right? So the DNA is in the cytoplasm, or it's just floating there in the cell, not really floating, but it will contain in the cell. And there was the need to develop these enfolding the plasma membrane, right? And that resulted in the endoplasmic reticulum, right? And then the nucleus, the membrane of the nucleus. And then what happened first is that this cell, now this eukaryotic cell that has a true nucleus, engulfed a bacterium, and that is an aerobic bacterium that has its own DNA. So this cell has the DNA here in purple. Now this bacteria has also, it's carrying its own DNA with it. And we have, as I have mentioned, mitochondria has its own DNA as well. Now, this cell with the mitochondria is an ancestral eukaryotic. Then this cell, or this type of cell, engulf a second type of bacteria, and that's a photosynthetic bacteria now, and that has resulted in the acquisition of this plastid. So this aerobic bacterium in time evolved into the mitochondria, and these photosynthetic bacteria in time evolved into the plastid. So this is the an ancestral photosynthetic eukaryote, and this is how these organelles evolve in eukaryotes. Again, we have a lot of evidence for that, and we're going to cover a little bit more of this next module. Plants have how many sources of DNA? One from the nucleus, two from the nucleus and mitochondria, three from nucleus, mitochondria, and chloroplast, from nucleus, mitochondria, chloroplast, and endoplasmic reticulum, or five from all of the above and the vacuole. Okay, so plants have a source of three: the nucleus, the mitochondria, and the chloroplast. Okay, that's the right answer. Now, I was talking about phylogenetic trees before, but what is a phylogenetic tree? This is the concept. It's an evolutionary history of a species or a group of related species. And what is going to be more important, or very important to remember, is that a phylogenetic tree is actually a hypothesis. And we want to test hypotheses. We propose this hypothesis, and then we want to test them. And that is our interpretation of how evolutionary relationships occur, or how the evolutionary patterns occur in time. This is a tree of life using Pokemon. And hopefully, if you like Pokemon, this is for the ones who like Pokemon. If you don't like Pokemon, just skip this. But hopefully you can find your favorite Pokemon and see if they are closely related to others that you are familiar, and you can look at the differences or similarities in different groups. So if we look over here, we will have these features in these Pokemon that are not shared in other parts of these trees. So this is just a game, but you could also look at this, the idea of the phylogeny, if you think about the languages. And I like this because many of you are bilinguals, or some of you are trilinguals, or maybe you're just starting a new language. So in here, we have a phylogeny that was built in 2011 to look at the language evolution. In my case, I speak Spanish. I'm from Mexico. That's my native language, or my native tongue. And I speak also French and English, but all of these languages here in green are descended from the Romance language. And for me, it's easier to understand Portuguese or French than, for example, German or Flemish over here. So look at this, German and English are more closely are more similar than what they are to any of these other languages over here. And some of you might be more familiar with Hindi, and you can see that Punjabi is also more closely related than what it is to German and so on. So hopefully you can take a look and find the language that you speak and see if that makes sense to you, that the closest language is in those branches. Okay, so now let's take a look more to these. Let's learn how to read phylogenies. In that phylogeny, we are going to have a common ancestor, and this is going to be a branch point that represents the divergence of two different lineages. In this case, we have an ancestor that lived in the past, and that is speciated, and that speciation led to this lineage that at the top has these fishes, or at the end is represented by this lineage of fishes. This is an evolutionary lineage, and if we zoom into this, we will have many species of fishes in this lineage. Now, this will be an ancestor in the lineage, right? We have species that evolve from other species that are represented in this branch. So these are many species that now have evolved and led to the evolution of frogs. In here, we have also an ancestor, and this branch point here is going to represent the sister taxa. This is the most closely related species to humans, are chimps, and that's phylogeny. And now I want you to find the common ancestor from lizards and humans, and if you are following, this will be the ancestors. The most common recent ancestor from lizards and humans is right here. Oops, right here. Now, if we look at this branching point, we see that lizards are sister groups of both, or a lineage that carries both chimps and humans. And frogs are the sister groups of these lineages, this lineage that holds lizards and chimps and humans all together. This is the whole lineage that is sister group of frogs. Okay, time is important in this phylogenies. When you're reading a phylogeny, you want to consider time. And this point of the common ancestor of all of these lineages has a most distant, is more distantly in time. So this is the past, and this is the present, okay? The other two concepts that I want to introduce is the outgroup. In this case, the turtle is the outgroup, and the in-group is all of these species that we want to study, the various species that we are interested on investigating. We have the class Mammalia. That means the mammals. Mammals are defined by the presence of hair, the presence of mammillary glands, the neocortex, and the 3 bones in the ears. Okay, so those are features shared by mammals. Now, we want to investigate mammals, and then we use this outgroup to actually understand the shared derived features in our phylogeny. So let's take a look at this. In here we have the presence of only one hair out of the 4 that I told you that are shared in all of the mammals. That's hair. Now, if we look at this order Carnivora, these 3 species, we see that they share having meat-eating teeth, meat-eating teeth. So those are carnivorous. And if we look at the leopard and domestic cats, both of them share this feature, which is that they have retractable claws. And if we only look at the domestic cat, this is the only one that is able to purr. So this is the direction in how evolution happened in this case. Time, the past is here at the bottom, and the most recent time is at the tips of these branches. So an autapomorphy is going to be a new morphological trait unique to a group in an evolutionary lineage. So in this case, for this lineage, domestic cat, the ability to pour, it will be our autapomorphy. The rooted tree, we're gonna use the outgroup to root our tree and have direction on this tree. So the turtle, as you can see, it's in one of the edges of the tree because we have also rooted the tree. Other very important thing is the way that we read these trees. And this is gonna be, hopefully you have a way to exercise your ability to rotate things easily, because this is a phylogeny that says that chimps and humans are more closely related. However, if we rotate this point right here and then we flip the humans and chimps, now imagine that if we only look at these relations, human and chimps are still the sister group in this phylogeny, but they have been rotated. Now humans are at the top and chimps at the bottom. So let's go over our next group. If we rotate this branch point right here, now lizards are gonna come down of this lineage, and that's what we see here. We have humans and chimps here, and lizards are at the bottom. If we look also at this rotation point right here, fishes were at the top, now they are at the bottom. So take a time and pause the video if you didn't follow and try to reconstruct what that means when we rotate the branches or the branch points. But what is going to be very important is, regardless of how we rotate it or we rotate these branch points, we are going to end with the same relationship, right? That means that humans and chimps, as in our original phylogeny, we're going to have, well, they're going to be still sister groups. And lizards are going to be the sister groups of this lineage that carries both humans and chimps, and so on. So this is important to consider. The other consideration is that there are at least two ways, no, there are more than two ways to represent the phylogenetic trees. One can be horizontal and the other one vertical or diagonal. In this case, we have this vertical tree. Again, chimps and humans were the sister groups, right? And in this diagonal tree, this is what we still have, is the same relationships with chimps and humans being the sister taxa. I want to make a parenthesis to say that this is what we think. We have an ancestor, a shared ancestor between chimps and humans, that from which these two lineages evolved. So people who say that humans evolved from chimps, they don't know what they are talking about, because no one in science have hypothesized that humans evolved from chimps, but rather then there was an ancestor from which these two lineages have evolved. Now, let's talk about systematics and the need of using different type of information. So we could use programs, mathematical tools, and computer programs to include or analyze sequences of DNA or morphology or other type of traits. In this example, we have 3 beetles, and these beetles are different in morphology. They are different species, 3 different species. In this example, we could have 3 evolutionary hypotheses. So if you don't know what the species is most closely related, you will have to investigate the different hypotheses. In this case, look at these beetles. These beetles, some of them look more bluish, others a little bit greener, and they have some spots here. This one has smaller spots, whereas this has larger spots. Based on this information, which one do you think is most closely related? What two species are the sister taxa? Yeah, you can come up with different alternatives, or with different options, different phylogenies and hypotheses. You could say maybe these two are sister groups because they have these spots in here, but maybe you could think that this is blue and this is a little bit bluish, so maybe these are more closely related. So this is what is being represented here. In this first phylogeny around here, we have that 1 and 2 are sister taxa, and then 3. And this phylogeny 1 and 3 are the most closely related, and then 2. And in this other one, 3 and 2 are most closely related, and with respect to species 1. Now let's imagine that we sequence a piece of DNA, a piece of a gene, and then we have these letters. So in this case, we also have a sequence from the ancestor. That is the ancestor from which this species evolved. The ancestor of these 3 species will be located in here in this phylogeny, in all of them, right here. And this is the sequence, CACG, CAGAGT, CT. And in red, we have substitutions that appear in each of these lineages. So we have sequenced these 3 species for this particular gene. And now what we need to make is a matrix. So if we are gonna do a matrix, we're gonna make a matrix with the variable size, the variable positions. And I'm gonna help you a little bit. So the first position is gonna be this one, the CCAA. And you want to find a matrix in the bottom that has these letters. The next position will be this, and this, and this. So please select the matrix that is constructed with variable size only. Okay, hopefully that was easy, super easy. I helped you a lot. That's this is the A option. Now, the next thing that we want to do, this is the matrix that we selected. That's the option A. The next thing that we want to do is to trace how these substitutions fall into these 3 different hypotheses. In this method, this is the parsimony, the maximum parsimony method. In this method, parsimony methods assume that the most simple explanation, or the most the explanation that requires the fewest steps, is the one that we should support. So in this case, maximum parsimony assumes that the tree that requires the fewest evolutionary events, and that means the appearance of the right characters, is the most likely. Okay, what does that mean? Let's go with an example to understand what that means. We have our matrix here. And what we need to do is to look at the ancestor. So the ancestral sequence has an adenine. That means that if we find the ancestor in the phylogeny, we're going to have an adenine here. Then species A, for site 1, has also an adenine, right here. So then this one is going to have an adenine here. So we're not going to mark a substitution. We're only going to mark substitutions that evolve after the ancestor. So species 2 and 1 have a cytosine. And then we only need one, marking 1, because we can understand that if the ancestor of both the species 1 and 2 have the mutation, one of the ancestors here, both of these species will have a cytosine. So we mark 1 substitution here. Now let's go to the next site. So the ancestral will have a G. So this has a G. So we're not going to mark the species 3 because it has a G as well. But then what happens with species 1 and 2? now they have a T right here. So species 1 and 2 have a T, so I'll mark a T here. And what happens with site 3? And the ancestor will have a T, so we'll mark a T in here. But now species 3 and species 1 will have an A. So this is the substitution that I mark here. And now, but we need to mark twice the substitution, right? In this lineage and in this lineage. And that will explain the presence of adenine in lineage 3 and lineage 1. And then for site 4, the ancestor has a T. So imagine we have a T in here, but now species 3 and 2 have a C. So we want to mark those substitutions in this lineage and this lineage, but not in 1, because 1 is similar to the ancestral. So in this first hypothetical tree, we will count how many substitutions we mark. So we mark 1 in here, and then we mark 5 more in here, with a total of 6 events. And if you pause the video and do the same example or the same exercise with the hypothesis number 2 and hypothesis number 3, you will see that these 2 options will have 7 events. In this method, in the maximum parsimony method, we will prefer the phylogeny that has the fewest steps or the fewest appearance of shared derived characters, and that is hypothesis 1. So in this case, we will conclude that species 1 and species 2 are more closely related. I hope that makes sense. And now, the best hypothesis for phylogenetic trees usually is the one that is supported with several sources of data. That means morphological data, genetic data. Now we use also niche modeling data and fossil data, or other type of data that we can include. So, and if all of the data supports the same direction, we tend to favor that hypothesis. So this is, again, this is the one that we will support under the maximum parsimony, the one that with the fewest. If you are following what I just taught you, you'll be able to answer this question. So in Japan, a group of researchers were trying to determine if all of the meat from whales was legally hunted. And only the whales from the southern hemisphere can be sold legally in Japan, not other type of whales. So what the researchers did was they went to the markets and bought different types of meat from different vendors. And then they went to the lab, ground the tissue, the meat, and extracted the DNA and sequenced a piece of a gene from the mitochondria and compared it against known sequences. So they have mink from the southern hemisphere. They also have mink or mink from the North Atlantic places, humpback, blue whale, and fin whale. So these are the references that they use to match if the sources from the markets were sold illegally or legally. Now, the samples marked as unknown are samples from the market. Based on the information that I just told you, please answer, what did the researchers concluded? A, all whales were caught legally. B, some whales were caught legally and others illegally. They didn't have conclusive results, or all of the above, or maybe none of the above. What do you think? What do you conclude based on this? Well, it's quite easy, right? So some of them, like these unknown mitochondrial DNA sequences from these samples, match the ones that can be sold legally from the Southern Hemisphere. However, everything in red here matches or is closely related to species of whales that are sold illegally. So B is the right answer. Some whales were caught legally and others illegally, sadly. Okay, these two are concepts. These are two concepts that are important when reconstructing phylogenies, when we're using these type of traits. These concepts are homology and analogy. Homology, we have talked about it briefly before. It is similar. These are two features or a group of features that are similar to share ancestry, and they can be phenotypic or genetic similarities. And if we have organisms that look similarly in appearance, usually the genes responsible for those appearances are going to be similar as well if the features are homologous. In contrast, analogous, we're going to have that the similarity of these features is due to convergent evolution. And what's that? So let's look at some examples to clarify that. Convergent evolution is going to occur when we have similar environmental pressures, and that is natural selection also acting on the traits and to produce similar analogous traits. And these are going to be adaptations that, for example, the Australian mole and African golden mole are going to be developing in similar environmental conditions. So these two species are distantly related. They are not from the same lineage, so they don't have the same most recent common ancestor. However, if you look at them, they're going to have similarities, such as reduced vision, these large claws to be able to dig on the ground, and this anatomic feature, right? So this has evolved as a result of living in similar conditions on the ground, but from different ancestors. So natural selection, again, has shaped these convergent traits. Another example are snakes and glass lizards. So these are legless. So they lack legs, right? The snakes and glass lizards. But you see, they are not that sister group. Actually, monitor lizards are the sister groups of glass lizards in this phylogeny. And what this means is that the absence of legs has evolved twice in this whole group. So they have evolved twice by convergence, not because they share the most recent common ancestor in between these two groups. Cladistics is going to be grouping organisms by ancestry. And in this sense, we are interested in defining a clade, and we want to define a clade that is going to be including the ancestor of the species and all of the descendants. If we have other configuration, we usually define these as unnatural or artificial groups. So we want to have these clades defining our hypothesis of a group. Now, we are going to have three possible type of clades. And the one that I really want to emphasize is the monophyletic group. That's the natural group. And when we see that in a phylogeny, we are happy. And a monophyletic group is going to be the one that has the ancestor with all of the descendants. I have these three phylogenies here. And if you look at the block in blue, you'll see that this ancestor is contained in this blue box. And it also has all of the descendants. So we have three descendants coming from this ancestor. That means A, B, and C. And this is a monophyletic group. A paraphyletic group is going to be a group that has the ancestor and some D, E, and F of the descendants, right? So it has some of the descendants, but not all, because it's missing G. Okay? And a polyphyletic group is going to have a species that lack or a group that has a species or the descendants that lack the most recent common ancestor. Right? So in this group, if you look at the most common recent ancestor of D is located right here, and this artificial group won't have the ancestor. This is an example of both artificial groups. That means a paraphyletic group and a polyphyletic group. In this case, we will have descendants of this ancestor. So the ancestor is the even-toed ungulates. But let's say that the group that I am interested, I name it the even-toed ungulates that are herbivores, and they are terrestrials. So that's the group that I think that's a natural group. So I want to test that in a phylogeny, and I find that it is not. And it is not because this group is not including the cetaceans, which is a marine, is not terrestrial. Right? And then that therefore will have, I will conclude that the group that I was testing included the ancestor and some descendants, but not all. Therefore, this is not a natural group, right? That's not a natural group. It's an artificial group that I have created because I probably don't understand how the evolutionary patterns occur. Another mistake that we can make is that we could look at convergent evolution and or conclude that we have a group that is actually an artificial group. In this case, we have cetaceans and seals, and we can call them that they are marine mammals. So are these marine mammals monophyletic? They are not monophyletic. They are actually polyphyletic because this group, these two species, actually the grouping is not including the most common recent ancestor for these species. That's an artificial group that evolved through convergence. Now, this is a phylogeny that includes the cocodrillians and dinosaurs, as most people call these dinosaurs. That means excluding the birds, and we will have here the birds. So the common ancestor of these four lineages, it's right here. All of these species have these common features, these shared traits, and that's the four-chamber hearts. They are or were able to sing or emit some types of sounds as a singing. They build nests, and they also exhibited brooding. And brooding means the behavior to warm the eggs. And we know that based on some cool fossils. If you look at this picture, this real picture of a fossil that has the eggs. Right here we have some eggs, and over here we have also more eggs. And these are the bones of probably the mother or maybe the father that was warming the eggs in this species, the oviraptor. Now, are dinosaurs a monophyletic clade? And I want to call dinosaurs in the sense of just these extinct dinosaurs. So extinct dinosaurs like the ornithischian dinosaurs or the Saurischian dinosaurs. These two lineages are actually paraphyletic because if we trace the most common recent ancestor, it will be right here. This is the most common recent ancestor of these two lineages, and they are they include some descendants, but not all, because they are excluding birds. And nowadays we understand a little bit more, and then actually birds are considered the modern dinosaurs. So if we use the word more inclusively to refer to all of these as a dinosaurs in a more inclusive way, you will conclude that these are monophyletic because they have ancestor here and all of the descendants. So label whether the groups highlighted in pink are paraphyletic. For example, in here, is this group paraphyletic? The archosaurs. So the archosaurs are highlighted in pink. Are they paraphyletic? No, they are not. That's false. They are actually monophyletic. Now, check true or false if these, the dinosaurs, as in the more strict sense, are monophyletic. Is this true or false? This is actually false. This is a paraphyletic group because it includes some descendants, but not all, right? So this is wrong. This is not monophyletic. Now, is this group in pink, the bats and hawks, these will be flying animals or flying vertebrates, polyphyletic? Yes, they are polyphyletic. They include the descendants, but not the most common recent ancestor of these two groups. So, right, the most common recent ancestor is right here, and the other one is right here for bats. So, no, they are not monophyletic. They are polyphyletic. So, they are examples of artificial groups. Now, if we want to reconstruct the phylogenetic relationships using DNA, we need to use the right molecule that is going to give us the correct resolution that we are using. In other words, if we are evaluating, for example, relationships between the 3 domains, we want to use a molecule of DNA or RNA that is able to, that it's conserved among all of the different species. That means that it exists there, it's present in all of them, but that it has enough changes to be able to disentangle what species it is which one. That means that mutations that can tell apart different species or at least different genera. In that case, we are using ribosomal RNA because it has changes that are relatively slow. But what if we want to be more, if we want to zoom into a particular group, for example, a group of animals? Well, maybe we want to look into the mitochondria, and that's because in mitochondria it evolves more rapidly, so we will be able to tell apart different species. Whereas maybe with ribosomal RNA, we won't have that type of resolution. So it really depends the type of question that we want to answer, is the type of molecules that we use. Nowadays, actually, we try to include all of the genome. So that means all of the sequences, all of the letters in the genome to reconstruct phylogenetic relationships. And that used to be very expensive, but it's becoming more affordable, and the resolution that we get is really high. So that's what we are doing currently. When we use the ribosomal RNA to create this evolutionary history or the relationships among the domains, this is the type of phylogeny that we get with the 3 domains, as I mentioned it before, the domain Eukarya, Archaea, and Bacteria. I want to introduce this concept of a polytomy. And a polytomy is not this bifurcated tree. Like in here, we have this bifurcation. So these 2 lineages coming from this branch point. And again, we have this branch point split in these 2 lineages. In this case, we have polytomy because we have these 4 lineages that are not bifurcated. Hopefully you can appreciate that this is a line that these 4 lineages are coming from the same position and at the same time. That's a polytomy. Now, the history, evolutionary history, early on time, during the origin of life, is very messy. And it's messy because a species, in particular in Archaea and Bacteria, they are able to have horizontal gene transfer. And horizontal gene transfer is the movement of genes from one genome to another. And this is an example, Galderia sulphuraria. It acquired around 5% of its genes from archaeal species and also bacterial species. That species grows in water, running water, that has really high temperatures. And that ability, those genes from archaeal species and bacterial species most likely resulted in these adaptations to live in higher temperatures. So this is a very cool example of horizontal gene transfer, the acquisition of genes from a foreigner species. And when we have that, that can result in this very messy pattern that we see in this more network type of representation. Instead of having this bifurcated phylogenetic tree, now we have more like a network, right? So in here, also we have the representation of the transfer from the mitochondria and the chloroplast. If you remember the endosymbiosis that I mentioned before, this theory eloquently presented by Lynn Margulis, we now know that the mitochondria is the result of the endosymbiosis. So those are bacteria getting engulfed into eukaryotic cells, and also that happened with the chloroplast. So this is like like an example of a massive transfer of information from one species to another. And in this case, is the endosymbiotic theory. I hope that you enjoyed this class. So now you know more about how we classify species and the methods that we use to reconstruct phylogenetic relationships. I'll see you next time, and please keep preparing for your exam. I'm going to be holding some office hours, extra office hours, so please come to my office hours. I'll be happy to explain any of the material covered today or in any other module before this exam. Thank you very much.